Today we will talk about white blood cells. White blood cells, these are granulocytes that have granules, neutrophils, eosinophils, basophils, and agranulocytes that lack granules, lymphocytes and monocytes. As we had already discussed, normal count ranges from 4,000 cells to 11,000 cells per cubic mm. About the percentage of WBCs in the circulation, around 50 to 70% are neutrophils, followed by lymphocytes 20 to 40%, monocytes 2 to 8%, eosinophils 1 to 4%, and basophils less than 1%. We will discuss about the characteristic features of white blood cells one by one. First, we will talk about neutrophils. Their diameter is around 10 to 14 microns, and the nucleus, it is of bluish violet in color, as you can see here, and it can be bilobed to multilobed. So these lobes are connected by the chromatin threads. Cytoplasm, it is slate bluish in color, and it has purple or pink colored fine granules. Main function of neutrophils, it is phagocytosis. So we can say neutrophils are our first line of defense. Then about eosinophils, diameter is same, around 10 to 14 microns. Nucleus, it is bluish violet in color, as you can see here, having 2 lobes and spectacle shaped nucleus. It is very important point to note here, spectacle shaped. Cytoplasm, it is light pinkish in color, but it has brick red colored coarse granules not covering the nucleus. About the function, eosinophils, they are anti-allergic, anti-parasitic, mild phagocytosis. It can also be their function. Next are basophils. Diameter is around same, it is same 10 to 14 microns. Nucleus, it is bluish violet and irregular type. Cytoplasm is bluish basophilic, and it has deep purple granules completely covering and distorting the nucleus. Mildly phagocytic activity is there, and they release heparin, histamine, serotonin. So there is promotion of inflammatory reactions as they release histamine, serotonin. Okay, they have a role in inflammation. About lymphocytes, they can be large lymphocytes. Diameter is 10 to 14 microns. Nucleus is bluish violet. It is single, can be central or eccentrically placed. Cytoplasm is bluish. A thin rim of clear cytoplasm is there, and no visible granules are there. Mainly they are involved in the production of antibodies and immune response. Lymphocytes, they can be small lymphocytes also. Diameter is lesser, 7 to 10 microns. Single nucleus, typical ink dot appearance. Cytoplasm, a thin rim of light blue color cytoplasm is there, and they also lack granules. About monocytes, these are the largest blood cells. As you can see, the diameter also 14 to 18 micron. Nucleus, it is pale bluish violet. Shape is bean shaped, or we can say kidney shaped. Bluish cytoplasm with no granules. Function, phagocytic function is there, and these are referred to as the second line of defense. And monocytes, when they mature into macrophages also, they can be a part of monocyte macrophage system. About leukopoiesis. Leuko means leukocyte, poiesis means formation. So the process of formation of leukocytes from the progenitor cells, it is called as leukopoiesis. Okay. About the stages of granulopoiesis. Process of formation of granulocytes is granulopoiesis. Process of formation of monocytes is monocytopoiesis. Process of formation of lymphocytes, it is lymphopoiesis. They start from primitive stem cell. It get differentiated into myeloblast, Myeloblast is converted into promyelocyte that forms myelocyte. It forms metamyelocyte. Here nucleus is horseshoe shaped. It will form the band shaped nucleus called as band form or juvenile form. Ultimately there is formation of mature granulocyte which enters into the peripheral circulation. Now how the process is regulated? It is regulated by negative feedback mechanism, in which whenever there is increase in the formation of granulocytes and monocytes, they exert negative feedback effect over bone marrow and there will be regulation of the process of leukopoiesis. Here are the conditions in which the WBC count is altered. First is leukocytosis, in which the WBC count it exceeds more than 11,000 cells per cubic mm, seen in case of infants, pregnancy, childbirth, exercise, pathologically in case of acute pyogenic infections, malignancies. Okay. Opposite to it is leukopenia, in which the WBC count is reduced, becomes less than 4,000 cells per cubic mm. It can be seen in case of known pyogenic infections, for example, typhoid and paratyphoid fever, protozoal infections like malaria, viral infections like smallpox, mumps, or in case of bone marrow suppression. Now about a very important topic, phagocytosis. What is phagocytosis? It basically means cell eating, in which there is ingestion of the invaders that attack the cells. And this is the main function of neutrophils and macrophages, which are first and second line of defense respectively. Okay, so the phagocytes, they ingest the foreign invaders, dead or the inactive tissues, with sparing of the natural tissues of the body. That means these are selective in nature. Okay. There are several stages of this process of phagocytosis. First is the stage of opsonization. Okay, in this stage, the microbes, they get coated with opsonins. These are the naturally occurring factors in the serum to form a bond or linkage between bacteria and the phagocytic cell membrane. Second is the stage of engulfment of the opsonized particle. Okay, so it is carried out by the formation of pseudopodia around the particle to be engulfed. Okay, pseudopodia envelop the particles and resulting in the formation of phagocytic vacuole. And once the lysosomes, they come into action, they fuse with the phagosome, resulting in the formation of phagolysosome. Then there is a stage of degranulation, in which the granules in the phagocytes, they get discharged. Okay, they get fused with the phagolysosome, and there is stage of degranulation, which ultimately ends up in the stage of killing of the microorganism. The bacteria, after being killed, gets removed or degraded by the action of hydrolytic enzymes. And this killing or degradation, it occurs by oxygen dependent and oxygen independent pathways. Now a word about monocyte reticuloendothelial system. Okay. So these are the cells that are included in monocyte macrophage system. The tissue macrophages in the lymph nodes, okay, Kupffer cells in the liver sinusoids, glial cells in the central nervous system, macrophages in the spleen and bone marrow, alveolar macrophages present in the alveolar walls of the lungs, mesangial cells in the kidneys,